All right, well, I'd like to welcome everyone to today's edition of the Polyploid webinar. We have two great talks today, and up first is Dr. Wendy van Drunen. Wendy is currently an insert postdoctoral fellow at Queen's University, working with Janice Friedman, where she's investigating evolutionary interactions between polyploidy and reproductive or life history strategies. She will soon be starting as a postdoc at the University of Toronto, where she'll be studying polyploidy in the context of urban evolution and ecology. And today she's going to give us a talk on the impact of urbanization on polyploid versus diploid plant species in Canada's largest metropolitan area. And I'm really looking forward to this. And with that, I'll let Wendy take it away.
All right, great. Thanks, Mike. So hi, everyone. I'm Wendy. I'm really pleased to have the opportunity to contribute to the Polyploidy webinar today. So I've greatly enjoyed it over the past year, so it should be fun. So today I'm going to be presenting some hot off the press results from some new research projects on polyploid evolution in urban environments. So I think this is maybe one of the first talks I've given where more than like 2 or 3 people in the audience actually know what polyploidy is. So I don't have to tell you too much about it, but we all know that in plants and in angiosperms specifically, polyploidy is particularly common. And we have notoriously invasive plants, maybe native plants that we can see outside our windows, and maybe many crop and horticultural species as well that are polyploid. So, while all angiosperms share at least one whole genome duplication event, polyploidy is not evenly distributed across angiosperm groups. Some families have rather rampant polyploidy, where others have very little. And nor are these polyploidization events evenly distributed throughout time. So there's been many excellent studies and reviews that have argued that historical whole genome duplication events tend to cluster in periods of drastic global change, such as the KPG extinction event here about 66 million years ago, which corresponds to the shaded area. We can see all these little whole genome duplication events in red there. And there's 3 things that are believed to be happening here. So 1, the environmental conditions during these events may actually be causing polyploidization events. 2, the short-term advantages of polyploidization are believed to contribute to adaptation during these periods and perhaps the preferential survival of polyploid lineages. And then the long-term advantages of polyploidy may be linked to eventual diversification. So in the present day, plants are facing a new challenge. So we have global change proceeding at an unprecedented pace. And one of the most extreme and rapid examples of these human-driven changes is urbanization. So evolutionary ecology in urban systems is driven by these abiotic and biotic environmental gradients. We have gradients in water availability or temperature, or perhaps pollinator reliability. So humans are creating these novel environments through the expansion of human settlements in built-up areas. And while we know a fair amount about urban ecology, aka maybe how these gradients affect interactions between species, we actually know very little about how urban environments affect species evolution, which could be in terms of selection or genetic diversity, and really ultimately the evolutionary trajectories of species in urban environments. Because researchers have really only started digging into the evolutionary side of things in the past 10 years or so. And this is particularly true for plant systems over animal systems. But in general, urban environments are excellent places to study rapid real-time evolution under severe and changing selective pressures. And even better, every city out there is actually a replicated experimental unit, and so is great for assessing the consistency of evolutionary effects or convergent evolution. So we might ask, if whole genome duplication events and polyploidy are associated with past environmental change, do the ecological and evolutionary conditions associated with urbanization also favor the persistence of polyploids over diploids? And while we know from some studies that polyploids can be associated with disturbed environments or maybe agricultural habitats, aside from some speculation, really nothing is known about the interactions between polyploidy and urban environments specifically. So let's say we have our typical busy urban area here. We've got some roads and some cars, some suburbs, some gardens, some construction and development, a dense downtown core, maybe a little park, and perhaps even some pollutant producing factories. So we can use our knowledge of polyploidy and how it may have worked historically to come up maybe with several hypotheses about what polyploids might be doing in urban areas now and how urban areas may actually promote polyploid evolution. So I'd like to briefly go through these 6 hypotheses that I have here, many of which are interrelated. But first, I'm going to pause and say that a lot of these arguments that I'm going to be making in the next couple of slides are very general. So as we all know, polyploids are nothing if not inconsistent, and the traits of naturally occurring polyploids are highly variable. And this can mask our null expectations of what polyploids should be like. And in the end, like almost everything else involving polyploidy, the behavior of polyploid species in urban environments is likely to be very species dependent. Okay, but first, so our first hypothesis here is that urban areas should actually promote the formation of both auto and allopolyploids. So stressful environmental conditions such as temperature fluctuations or chemical pollutants are known to influence and reduce gamete production, and this should directly affect the rate of autopolyploid formation. And urban environments also bring together many species that otherwise would not be in contact in nature, and this increases the potential for hybridization and the formation of allopolyploids. And notably here, recurrent formation of neopolyploids in urban areas may refresh or sustain urban polyploid pockets of plants, regardless of actually any ability of those plants to survive well in urban environments. Maybe a key point to keep in mind. But our second hypothesis might be, so theory and minority Cytotype exclusion principle suggests that newly formed polyploids are going to have the most success establishment when they're able to colonize new habitat away from their diploid parents. Now, because urban habitats are very heterogeneous, we have maybe some sparse suburbs versus built-up urban or downtown areas, and this heterogeneous nature of urban areas may provide many opportunities for polyploid establishment. And the potential for novel traits in neopolyploids may further facilitate establishment if they have shifts to new niches. So while this, I've been talking about this in the context of new polyploids, this could also be applicable to established polyploid species if they are better able to colonize this continually opening urban habitat better than diploid species. Maybe they are better competitors or more tolerant of urban conditions. Which brings me to hypothesis 3, in that polyploids are often considered as having broader ecological niches than diploids and being able to withstand more stressful conditions. Whether or not this is actually true is another story, but we might have changes in physiology between diploids and polyploids that alter responses to drought, mitigate water loss, affect photosynthetic rates, or affect the ability of a plant to deal with a saline environment, all of which are key in densely urban environments. And this may eventually lead to polyploids outcompeting diploids in urban areas. So number 4, polyploidy may actually facilitate rapid adaptation in response to urban evolutionary pressures. So polyploids in general have a greater effective population size. They have higher heterozygosity than diploids because they have more chromosome copies, and they might have higher genetic or enzymatic diversity and perhaps even the potential for genetic novelty via neo or subfunctionalization. And again, this is variable for polyploids across the board. But particularly for new polyploids that are just being formed in urban environments, they may be able to kind of sample phenotypic space away from their parents and exploit new adaptive peaks in novel urban environments. So polyploidy is also associated with various forms of reproductive assurance. This could be things like apomixis or clonal reproduction, self-fertilization, self-compatibility, or perenniality here. So, and polyploids with their reproductive assurance and maybe even low inbreeding depression may be better able to withstand unreliable sex in urban environments better than diploids, which may be due to unreliable pollinators in urban areas or a lack of resources to put into sexual function. And moreover, an increased reliance on asexual reproduction or selfing in urban areas is actually predicted to further increase the production of unreduced gametes in these species, again, potentially promoting the formation of new polyploids in urban areas. And lastly, human activity may actually kind of predispose urban areas to having polyploids, regardless of any advantages that those polyploids might actually have. So urban areas have high proportions of alien and invasive species, many of which are polyploid. They also have high numbers of agricultural plants and human commensals, many of which are again polyploid. And even our horticultural or landscaping choices in urban areas can affect polyploid occurrence. So we have our number of hypotheses here on urban polyploidy. And what I'd like to do today is not actually specifically test any of these, except maybe intervention. So thanks COVID for restricting me to computer-based studies. But that's coming up next in the fall during my next postdoc. So today, what I'd like to ask is, if any of these hypotheses are in effect, what patterns should arise in urban areas? What sort of patterns should we see between species in urban and rural environments? So many of these effects might be more apparent thousands, if not millions of years from now. But urban environments present extremely strong selective pressures, and they can lead to very, very rapid changes in plant communities through ecological environmental filtering in urban areas. So while I can't address anything to do with the formation or establishment of new polyploids within species, I can ask, can we see any interactions of urbanization with existing polyploid and diploid species right now? So I'm going to present a few exploratory analyses aimed at addressing these three questions. So I'm going to ask, are polyploid species more common in urban versus non-urban areas? Are diploid versus polyploid species distributions changing throughout time? And are polyploid species associated with stressful urban conditions? So to do this, I'm going to use a few different data sets from the city of Toronto and the Greater Toronto area. So Toronto in Canada is Canada's largest urban area and contains approximately 15% of Canada's total population. So while Canada is kind of small, maybe on the global stage of urban environments, and it's a relatively young city, it is the most densely urban area in Canada. Okay, so the first question, are polyploids more common in urban versus non-urban areas? This was the focus of undergraduate thesis student Ida Mortazavi this past year, who I had the pleasure of supervising. And she went through and collated species observations from 30 parks in the Toronto area that were from data collected by the Toronto and Region Conservation Authority. So she took the species observations from these parks, and the parks were classified into three types. We have urban, rural, or something in between. And these are based on location as well as park use throughout the area. And then Ida went through and took all those observations and found chromosome numbers for all the species. She assigned them ploidy levels and a few other traits using the chromosome count database and independent literature searches. And I should note that we decided to restrict the data to only angiosperm species, and we also omitted most of the Cyperaceae that occur in the area due to difficulties in ploidy assignment. So overall, in the end, Ida's dataset contained over 9,000 observations of about 1,200 species. And interestingly, Ida's main result was that she found that the proportion of polyploid species in urban parks was significantly higher than the proportion of polyploid species in rural parks. And these are analyses using binomial families and generalized linear mixed models. So we have this rather clear result for urban versus rural parks. And it seems that this pattern does not appear to be due to a high number of introduced or invasive polyploids in urban areas. So while introduced species also increased with urbanization, they were not much more likely to be polyploid versus diploid. And the same for, or sorry, not the same for invasive species, but that we actually see no difference between invasive species between rural, urban, or in between parks. And there was no difference in the probability of a diploid or an invasive or non-invasive plant to be diploid or polyploid. And so when we go even a little bit further and we restrict the data set to only native species, we actually see the same pattern still. So that rural parks contain a lower proportion of polyploid species than urban parks. And if we look at the numbers themselves that are behind all these proportions, we can see that these trends are driven by sharp decreases in the number of number of diploids from rural to urban sites, and not necessarily in the number of polyploid species that are occurring there. So there seems to be some support for an association between polyploidy and urbanization in this first project that appears to be mainly maybe driven by a loss of diploid species in urban areas. Okay, so let's move on a bit and ask whether diploid or polyploid species distributions are changing over time. So to answer this question, I took advantage of the TRCA's long-term monitoring plots program. So these are 40 fixed plots throughout the TRCA jurisdiction, and they've been sampled each year for species occurrences from 2008 to 2020. So these plots are again classified this time by the TRCA into urban versus rural plots and wetland versus forest. So I'm just going to talk about the forest data because the wetland data isn't any different. Well, we have 12 years worth of data here, over 20,000 observations, and over 500 species that are found in these areas. But overall, we see no effect of plot class. So there's no differences between urban versus rural plots in the proportion of polyploids that are found in them. And there's also no change over time. So, well, the absolute number of polyploids and the absolute number of species in the plots may be changing. There's no difference in the proportional number of polyploids that are at these sites. And this is perhaps not surprising. So 12 years and very recent years of that is not a long time for ecological filtering to occur to any extent. And in comparison to the parks data we were just looking at, where we have kind of the final product from the beginning of development in Toronto until now, this is just a relatively short snapshot. So let's go back briefly, maybe to one of the parks. So Hyde Park is a large urban green space. It's very close to the downtown core. Here, our downtown core is shown by this red star there. So fortuitously, there's detailed data on species extirpations in this park going back to the late 1800s. So I was thinking maybe we can ask, are diploid species more likely to have been extirpated from the park than polyploid species? And among native plants, the answer is maybe. If you squint a little bit, it looks like there is a larger proportion of diploid species that are being extirpated through the years than polyploid species. But we see no overall changes in community composition in regards to the number of polyploids or diploids that are found in the areas when we compare historical, so pre-1950 communities to what's in the park today. And I must admit, so this might actually be a pretty bad example to look at. So one thing, we don't know anything about species introductions in these areas, so we know about extirpations, but it's not necessarily the case that all native plants that are in there now are actually historically found in that area. So there's a bit of confounding things going on there. And High Park in general, it actually contains a very rare ecosystem type in southern Ontario. It's called oak savanna. And that means that High Park is very actively managed by the city, including active burns and general conservation of the species that are there. So this actually might be really negating any shifts in community composition that might occur between diploids and polyploids under less strict management involvement. But overall, with these two data sets, there doesn't seem to be much changing in regards to polyploid composition over time. So let's go a little bit further and ask whether polyploid species are more likely to occur in stressful environments. So to start, first thing we're going to look at is whether polyploids are found closer to the urban core than diploids. So here's our urban core, the red star again. It's Young Dundas Square if anyone is interested. But to answer this question, I'm going to use research grade observation data from iNaturalist, observations from 2015 to 2020 in the Toronto and Greater Toronto area. So we have about 18,000 observations and 8,500 species that this data set encompasses. And so we want to ask whether the polyploids in this data set are more likely to be found closer to this red star than the diploids. And across all observations in the data set, the answer is a resounding no. So there's no difference in the distance, average distance of these species occurrences to the urban center. And the same is true when we look at native species only. And if we look at species means across all these observations, we also see no difference. So this p-value is from the ANOVAs here. And I actually threw in some phylogenetically corrected analyses using PGLS. But there is still no difference to be seen in where diploid versus polyploid species are occurring in regards to the urban center. So the last thing I did was take kind of an independent contrast approach using sister species pairs that differ in ploidy. We have 61 pairs across the whole data set, and then within native species, we have 18 pairs. But again, we see no effect of ploidy in the distance to the urban center, but there are significant differences between species pairs, some of which do show that polyploids occur significantly closer to the core, but there are some that show the opposite effect as well. Okay, but really, even though distance to the urban core is a frequently used metric for urbanization, it might actually be a fairly poor measurement because urban areas do not get progressively less urban in a nice smooth way as we move away from the core. So let's be a little bit more specific and ask whether polyploids are associated with hotter temperatures in the Toronto area. This is known as the urban heat island effect, where temperatures within urban areas are usually significantly warmer in the summer and significantly colder in the winter than non-urban areas. So again, I'm going to be using our set of research grade observations from iNaturalist for this. All our little black points there. And we're going to map those observations onto a layer of average land surface temperature, which here is the average for June in 2005 at 10 a.m. And this was provided from Natural Resources Canada from the Geological Survey. So if we look at temperature differences across all the species in our data set, we find that there is actually a small significant difference in the temperature that these species are occurring at, and the polyploids are occurring at a slightly higher temperature So the biological importance of that slight of a temporary difference is questionable, and we don't see that for the natives either. But if we look at the species means again, we see no differences, either the non-phylogenetic analysis or the phylogenetic analysis. And for the species pairs, there is again no effect of ploidy, and there is also no real difference between the species pairs in this analysis. So overall, polyploids are not occurring in generally hotter areas than diploids. And the one last environmental data set that I want to look at is impermeable surface cover. So impermeable surfaces refer to surfaces that cannot absorb water, like concrete or asphalt, and this is another common urbanization metric. And in this data set is a binary state. So these plants are either occurring on permeable surface or impermeable, and this is classified using satellite data, I believe. So again, we're going to use our iNaturalist data. We have our number of observations and number of species over here. And this surface cover data was provided by the City of Toronto. Okay, so across all species, polyploids were very weakly associated with impermeable surfaces, okay? So we're just looking at the number of species that are found on impermeable versus permeable areas, but there is no difference in the response of polyploids to these surfaces. And so next I calculated the probability of each of these species occurring on an impermeable surface. And again, we see a very weak association across species, but no association within native species. And we look at our sister species pairs. There is no consistent effect of ploidy, but considerable differences again across species. So in summary, so there are the first pass to this sort of problem, there might be a few intriguing patterns. So we found that urban parks contain a higher proportion of polyploid species, but we found no evidence for any short-term temporal changes in polyploid versus diploid occurrences. And we have maybe some weak associations with stressful conditions if we squint a little. But really, this small set of studies has just skimmed the top of the potential urban polyploidy relationship, and there are many next steps to go, even with these data sets that I've shown you now, including analyses of community ecology, correcting for differences in temporal and spatial sampling. We can include additional traits. We're just looking at polyploidy here, but polyploidy, as we know, is correlated with all these things like self-fertilization, which might be a better predictor of some of these environmental conditions or where species are occurring than polyploidy itself. And we also need to extend these tests to other urban areas. So Toronto is a relatively young city, and it's less dense than many urban areas worldwide. And lastly, of course, we need to have some empirical tests of what exactly is going on with polyploids versus diploid individuals that are occurring in urban areas. So I just wanted to briefly end with this quote from Gustafson from his 1948 paper. So he says, "Man is an active agent in transforming the natural floras, and polyploids profit more or less from his influence." This is a bit of a lesson again that researchers past have often thought about our new ideas now. And I also wanted to say that if any of these hypotheses that I outlined at the beginning of the talk actually play out, what does that mean for urban evolution itself? So aside from the effect of urbanization on polyploidy, what is the effect of polyploidy on urban evolution? And maybe future evolutionary patterns across the angiosperm species as a whole. So we know polyploidy has shaped plant evolution in the past, and if indeed polyploids are more common in these anthropogenic environments, we could be having a big effect on the role of polyploidy in the future in terms of the adaptation to global change and even speciation rates. And by actively considering polyploidy, we might get some unique insight when looking at adaptation and the effects of urbanization on plant species. So with that, I will end and just thank my lab group here at Queens and Ida, who really carried the first data set that we were looking at there. Mark Johnson, who is going to be my supervisor coming this September. He's been very supportive of my crazy ideas involving urbanization and polyploidy, and our various funding and partners. And I'd be happy to take any questions.
All right, well, thank you, Wendy. We're running right into the time for the next speaker, but we'll go ahead and we can take a question or two, I think, as we make the transition. And so if folks want to put their questions in the chat box and then turn on, maybe as Thomas, if you want to go ahead and fire up your audio, video, ask away. Well, I think that we can't hear you, Thomas.
Okay, well,
on the fritz.
There you go. There you go.
Thinking, like with the correlates of polyploidy and stress, I was also wondering if in these urban parks you think that some intentional planting may have occurred. Like I know you looked at also like invasive species and things of that nature, but some of the natives may have ostensibly been planted as well. Do you think human selection has also directly affected any correlates with urban environments?
Yeah, I think that's definitely possible. But there's not really much I can say about it now. I mean, it's a complicated problem. Obviously, this is just really broad level stuff at the moment. There's going to be so many different complexities, like when we actually go to a smaller level here. But yeah, I definitely think that what humans are doing in these environments may play as big a role as how the polyploids themselves are actually responding to it.
Yeah. Thank you.
All right, and then I see we'll take the next question here from Pam, and then we'll wrap up if Pam wants to pop on.
Hi, Wendy. Nice talk.
Hi.
Good to see you. I was wondering if you see any phenological differences or if you've started to look into that between ploidies across those parks as well.
No, I haven't looked at that myself. I know there are a lot of studies that have looked at the effect of urbanization and other things on plant phenology, but I don't know anything about that in this data set at the moment now.
Okay, great. Thanks.
All right, well, thanks everyone. And thank you, Wendy, for a great talk. We'll have time for questions at the end. So if anybody has any other questions for Wendy, feel free to save those till the end and we can all come back and ask both speakers questions then. All right, with that, I'd like to start our next speaker off here is Shelly Gaynor. I think many of us know Shelly from the excellent work that she's done as a student rep at the Botanical Society of America. Shelly is currently a PhD candidate at the University of Florida with Pam and Doug Soltis, where her work focuses on the formation and persistence of autopolyploid populations. And Shelly's going to tell us a lot more about that today in her talk today on modeling the dynamics of multiple origins and gene flow in autopolyploids and their diploid progenitors. With that, I'll let Shelly take it away.
Thanks, Mike. So I'm, of course, talking about what Mike said, modeling the dynamics of multiple origin and gene flow in autos and their diploid progenitors. And this is a lot of the work that I've been working on with Jose Miguel, a statistician at UF. And so just to start off very simple and make sure we're all on the same page, polyploids are often classified into two extremes. Allos, which have chromosome copies from two genomes, and these often experience disomic inheritance and bivalent pairing. And then we have autopolyploids on the other extreme, which can be derived from a single individual. However, they can also be derived from two closely related individuals within one taxa. These are expected to have polysomic inheritance and multivalent pairing. Though hybrids are quite cool, my focus is autopolyploids. Early polyploid researchers like Stebbins initially thought that autopolyploids were evolutionary dead ends, which led to less research in autos. Soltis et al. 2007 and Barker et al. 2016 have since argued the importance of autos. However, this lag has still caused less to be known about naturally occurring autopolyploid populations. And so, just to stress, autos are significant. They're common in nature and economically important. For example, major crops such as potato, alfalfa, and blueberry are all derived from autos. When an auto or an allopolyploid forms, they experience minority cytotype exclusion, as mentioned in the last talk. And this is negative frequency-dependent selection. For example, this newly formed tetraploid, or my purple dot, is surrounded only by diploids, the blue dots, and it has no one to mate with except itself. To escape minority cytotype exclusion and persist, simulations of specifically autopolyploid populations have shown that establishment can be aided by the divergence or expansion of phenotypes, as well as change in niche. And we could have shifts in reproductive assurance. There also might be spatial segregation and recurrent formation, and these have all been theoretically related to the establishment. I'm going to go through these a little more detail in the following slides. So first, divergence or expansion in phenotypes. This is often seen in both autos and allos. And here's a review that we published last summer on secondary metabolites broadly in polyploids compared to their diploid progenitors. We found no consistent trend. Some had no association with ploidy level, and some had novel metabolites at higher ploidy levels. Some even had fewer compounds in higher ploidy levels. So overall, there was no consistency, and this has been seen in many trait-specific investigations into the consequences of polyploidy. So zooming back to just autopolyploids, we also see a variety of trends when looking at climatic or ecological niche. In study on autos, we can see niche expansion, where the autotetraploid has significantly wider niche breadth than the diploid progenitor. There could be niche contraction, where it has decreased niche breadth, and it occupies a subset of the diploid progenitor's niche space. There could just be niche conservation, where it occupies the same niche as the diploid, or complete niche divergence. And these have been observed in naturally occurring autopolyploid populations, and in case you were interested in specific examples of autopolyploids with these niche dynamics. And like everything polyploidy, these are just the most clear-cut dynamics. For example, in Galix usulata, for our 2018 paper, we found climatic niche contraction and slight niche shift. So it looked a little like the plot you see on the side and isn't really as clear-cut as these other examples. Next, reproductive assurance. An autopolyploid may experience shifts in mating system to facultative, where you can have both asexual and sexual modes of reproduction. And this is thought to help with establishment because it will increase the probability of establishment. In addition, selfing could be done through the breakdown of self-incompatibility, which has been seen in empirical examples like Sutherland et al. 2018 on Campanula rotundifolia. Next, spatial segregation, which is a mechanism that creates a prezygotic barrier between cytotypes, thus allowing the newly formed cytotype to escape minority cytotype exclusion. It's expected that dispersal probability decreases proportionally with distance. Thus, this would allow establishment. So if a newly formed autotetraploid moves or originates in a peripheral region, gene flow should be decreased, and the cytotype may then experience a niche shift, further distancing itself from the progenitor. Spielhoff et al. 2020 supports the creation of a local majority in a region of narrow habitat to aid in establishment. And an even more recent study this year by Griswold modeled the adaptation to peripheral regions and found high pollen dispersal rate allows the polyploid to outcompete the diploid in peripheral regions, specifically when the genetic basis of adaptation is recessive. So moving on to origins and formation, when a new autopolyploid lineage initially forms and faces minority cytotype exclusion, the number of starting lineages may contribute to its success. Very few autopolyploids have been found to have a single origin. For example, we see this in Tolmia. However, alternatively, you can have multiple lineages from separate whole genome duplication events. Theoretically, repeated formations are thought to aid in the establishment of autopolyploid lineages since they add additional diversity or mates and provide multiple chances at establishment. These lineages could come together at some point, allowing the evolutionary histories to converge and be linked. Though we aren't sure if this is always occurring, we often treat autopolyploids as connected lineages. We have seen multiple origin more widely and naturally occurring autopolyploids, including Heuchera and Gametes Cuciolata. When thinking about these new lineages, here I show a simplified figure from Badul et al. 2018 showing the polyploidy hop, or the evolutionary trajectory of autotetraploids and their diploid progenitors. So following autopolyploid formation, I just talked about the changes that could be due to escaping minority cytotype exclusion. In addition to these, there are gene level impacts. These could be challenges during meiosis, cellular mismanagement, and gene dosage imbalance, which all need to be overcome. There also may be relaxed purifying selection, which causes the accumulation of TEs. At a population level, we expect high heterozygosity, low inbreeding depression, high mutational load, reduced linkage, and these dynamics can allow for adaptive advantage. However, it's really unknown if these dynamics cause consistent consequences among separate origins, and if separate origins escape minority cytotype exclusion in uniform manners. And this brings me to my centralized question and what drives my dissertation and my research interest. And I'm interested in knowing if these independent whole genome duplication events have distinct evolutionary trajectories. But before I could really address this, I need to make sure that I can identify origin events when gene flow is ongoing. So can the effects of multiple origin and gene flow be differentiated in a mixed cytotype autopolyploid system? I came to this question because Gametes Cuciolata has 2x, 3x, and 4x cytotypes. It ranges through the southern Appalachians, and there's an estimated 47 whole genome duplication events and 31 triploid events. There's ongoing gene flow between the cytotypes, which perhaps could homogenize separate origins and cause origin events to not be detected. So this could be an underestimation of the number of events. Alternatively, gene flow could confound origin estimates, since the tetraploid may be more related or seem more related to the diploid which secondary contact has occurred, rather than the diploid progenitor from which it came. So that could lead to this to be an overestimate. We often infer origin based on things like this tree, which is a neighbor joining tree based on Lynch distance, and then Chromadeval was used to predict origin events. The black stars indicate tetraploidy events, and the red stars indicate triploid formation. The shading just corresponds to a structure analysis of the diploid populations. This is just one of the many methods used to detect number of origin. Others use trees but with different distance matrices. They could use principal component analyses, structure, and various other analyses to determine if there had been multiple origins. But it's really unknown if any of these methods are ideal for detecting multiple origins in the presence of gene flow due to the lack of data with known histories. Ideally, to evaluate these methods, we would want to simulate diploid, triploid, and tetraploid gene pools where there's unidirectional gene flow from the diploid to the tetraploid. The triploid would form from 2x and 4x gene pools, and we would expect the 3x to flow into the 4x gene pool. However, triploids are complex and they could also flow into the 2x gene pool. And so for simplicity's sakes, I'm not going to discuss the triploid anymore in this presentation. So to break this down even further, what we'd want in the simulation is a 2x gene pool with disomic inheritance, population growth, a mixed mating model, so allowing selfing and outcrossing, with a certain number of generations before our polyploid formed. We then have our tetraploid formed through sexual polyploidization, which is the formation with two progenitors with unreduced gametes, which is thought to be more common. We then have our 4x gene pool with unidirectional migration from that 2x gene pool. And we would, of course, need initial selfings, so that's why we have this mixed mating model. And we'd allow population growth as well to a carrying capacity based on the length and width of a habitat. Now, when you go to the available autopolyploid models and you're trying to run something like this, there's nothing. Most models are made with unshareable code. Many of this is proprietary due to the use in crops. So you're really unable to build upon the work of others. Many models do not explicitly incorporate autopolyploid specific gametic probabilities. They don't allow for multiple origins. Instead of tetraallelic loci, they're just biallelic. And due to our marker availability up until recently, a lot of models are phenotype specific. There may be new models that can accomplish these things, but to my knowledge, there isn't. And so this brought me to making Autopop, which is an R-based autopolyploid population genetic simulation. It's R-based, so it's in a language that we all use. It's going to be open access and it will be available on my GitHub under mgaynor1/autopop. And this is to allow others to develop the model further. It's also parallelized to run locally and efficiently. And I'm excited to show you more. So this is a very messy diagram of what this model includes. And I'm going to zoom in and walk you through the bottom, which is our tetraploid. The top is our diploid in blue. So the first step is to go from our genotypes to gametes. So we use a transition matrix, which is the probability of each gamete forming from the parental genotype. We use this probability matrix to sample using a multinomial distribution to take that genotype and generate its gamete. Now, we want this transition matrix to be informed by autopolyploid specific dynamics. So we want tetrasomic inheritance, both bivalent and tetravalent formation. We want double reduction and partner pairing switch. So we all know, or probably have seen this tetrasomic inheritance Punnett square, where if you have the parental genotypes, you can get the frequencies that you would expect within the offspring. However, it's a little more complicated. At metaphase, you can have bivalent or quadrivalent formation. Quadrivalents are sometimes also called tetravalents. And these forming could then lead to different configurations of gametes. So here is a figure adapted from Ronforth et al. 1998. And in the first box here, we have those bivalents. And in the second and third, we have those quadrivalents. At anaphase 1, you could have reductional or equitational separation. And so in this first box here, you have reductional separation with no crossover between the centromere and locus. While in the second and third box, you do have crossover, but first equitational separation and then reductional separation. In anaphase 2, it's just the opposite. So if you had reductional in anaphase 1, it's equitational. If you had equitational in anaphase 1, it's now reductional. And you can see this gives you a diversity of tetrads, including 2 here that have double reduction and double copies of certain gametes or certain alleles. Now, when you go through all these pathways in a 2 loci tetraallelic parental gamete, you get 136 possible gametes. And this was derived by Fisher in 1947. So again, we have 2 loci, so we have A and B, and each of those have 4 alleles. Fisher then took this a step further and binned these in a biologically meaningful way. So first, if we had double reduction of A and B and 0 or 2 crossovers, we get 4 combinations in G1 and 12 combinations in G2. If you had double reduction in just A or just B with 1 or 2 crossovers, you get 12 different combinations in each bin. Now, if you have no crossovers or no double reduction and various number of crossovers, you get bins G7 through G11. And this is great, it gives us the combinations, but from here, there's a lot of work to be done because frequency doesn't equal probability. This doesn't directly relate to the probability of each of these combinations, just the pathway to get there. And so lucky for me, since 1947, work has been done to derive those probabilities. So in 2013, Resmuir derived the probability of each gamete based on 5 parameters, which I'll show in the next slide. And then in 2017, Griswold and Williamson built a transition matrix for biallelic possibilities. which is great. But I want those tetraallelic possibilities. And so instead of a smaller, more feasible matrix, this now expands our matrix to have all 136 gametes included and 9,180 potential parental genotypes. So this really expands our matrices a ton. So in this first step of our transition matrix, so given a parental genotype, what is the probability of each gamete forming? We have a diploid matrices of 10 rows and 4 columns that's based on recombination rate alone. And then we have our autotetraploid, which is much bigger, and that 9,180 rows by 136 columns. It's based on effective recombination rate, or Q, R, which is the effective recombination rate between locus A and locus B, P, PC, or the probability of paired partner switch between the centromere and locus A, and P, DP, the probability of a paired partner switch between A and B loci. We also have tau, which is the probability of tetravalent formation and allows both tetravalent and bivalents to be formed. So we take our parental genotypes, we sample these probabilities based on a multinomial distribution, and this gives us our gametes. These are not quite ready to enter the gene pool because we then take our gametes and we sample a mutation matrix, which is the probability that a mutation during the meiotic process causes a gamete to transition to another state. Again, the diploid matrices is quite small. It's 4 rows by 4 columns and is just based on mu, the mutation rate. For our autotetraploid, this is 136 rows by 136 columns, again, just based on mu, the mutation rate, but having these tetraallelic combinations or things that could happen makes this way more complex and was quite difficult to derive. So again, we sample this with a multinomial distribution, and then our gametes are ready to enter the gene pool. Because selfing is quite important for establishment, we allow a proportion of random individuals only to self, and this is done with sampling without replacement. The remaining individuals are randomly paired from the gamete pool, allowing outcrossing, and if there's an odd number of parents, we sadly throw one out because within each generation, we only allow 1 offspring per individual. So we take these offspring genotype and we make I equal to 1. And we take our previous generation genotypes where I is less than a set size and add these together for our parental genotype pool. So what is I? We're assuming perennial herbaceous, and I is by size and not age. And when I mean size, I really mean reproductive ability. So we know that autopolyploids will have a longer time to reproductive maturity compared to the diploid populations. And this is where we can incorporate that idea. And we also will then have a carrying capacity that's predefined. And this is based on work by John Spielhoff published in 2020. We also have migrants from the 2N gene pool at rate of M, and they're randomly sampled unreduced gametes. So now when we put this all together, we go back to this original map of my simulation. On the top, we have those diploids where our parental genotypes, we take those through a transition matrix, we get those gametes that are not yet ready to enter the gene pool. We go through a mutation matrix to see if it transitions, and then those gametes enter the gene pool and can self or outcross, yielding our offspring gamete that go back into that parental gene pool. We then have migration rate from diploids to tetraploids of a M rate, and it's a random sample. Our loop for our tetraploids in purple on the bottom is really similar, so I will not walk you through that. So in summary, currently I'm assuming unlinked loci and no selection. Our diploid source population has random mating, dysomic inheritance, population growth, a mixed mating model, and a stage structure model, or that I value. Our autotetraploid population will have various numbers of origins from single diploid population with temporal variation. We also have multivalent segregation, double reduction, mixed mating model, stage structured model, and various levels of unidirectional gene flow. So just to show you a simulation, here is our diploid with 100 individuals for 1,000 generations. This does not include population growth. Here we have A1 in red and A2 in blue, and because this has selfing, as well as outcrossing, we would expect a pattern like this. There's no fixation of A1 or A2, and it's quite stochastic over the generations. So this is what we expect. Now, since my question wasn't actually just, can I run a simulation, and it's testing those methods and the validity when looking at multiple origins in a population that has gene flow, hopefully by Botany 2021, I'll be finished with those new analyses and be able to share what I have found. And with that, I want to thank my committee members and collaborator and funding. And if you're interested in collaborating or expanding these models, please reach out, and that is why my email is up.
All right, well, thank you, Shelly, for a wonderful talk. As always, if folks have questions, we'll go ahead and pop those in the chat box, and then feel free to put your audio and video on. I was just going to comment that I do look forward to seeing the matrix size for the autododecaploid version of the model.
It's crazy looking. I could show it to you.
I'm sure. All right, lots of congrats there. I'm curious, real quick. Oh, Brittany, you have a question?
Go ahead. I'm still formulating a question.
I'm in the middle of that too. I was sort of curious though how sort of flexible you can be with. It seems very, very flexible with in terms of incorporating that switch from tetrasomic to disomic inheritance. And that was something that in the work that we were doing with Paul, for developing empirical sort of models of demography and autopolyploids was something that was a real challenge, right? Because we don't really have great estimates. It's hard to go out and estimate that necessarily for any non-model species. And I am sort of curious how easy it will be to incorporate, or it seems like you've already got it in there, but to incorporate sort of that shift from disomic to tetrasomic, and if you can apply that to different sets of linkage groups or chromosomes in your simulation, so that this set of chromosomes is still tetrasomic and this set's disomic, that kind of thing, or if it's more or less just everything is along some gradient from 0 to 1 of tetrasomic inheritance, or whatever. If you could talk about maybe just a little bit more about how you're thinking about that.
So with the tetravalent and bivalent formation and that kind of shift in allowing, to shift away from what we typically expect, it's part of the 136 gametes, disomic inheritance related gametes. And so we could just shift that by that probability. And so that's really just taking a probability of tetravalent formation. And so it's not like a 0 to 1, it's not binary, it is allowing kind of a spectrum to occur. So we can take empirical data and use that to inform the model there. It's a really cool way to look at the probabilities of these gametes. And that's really not what I've done, it's what people have done before me and what that Resmir 2013 paper did. But it really does allow for a spectrum rather than to say like discretely it's occurring or not occurring. So you could use it to inform different areas of the genome. Currently, I'm like simulating it with haplotypes rather than specific sites. And so I could change that to do that as well if that's something that fits questions out there.
Yeah, very cool. I look forward to seeing. Poly's a different approach, at the end of the day, to estimate it and put it in this, because we're looking with site spectrum data, so it's a little bit different sort of approach. But I really like what you've got, and I'm just sort of thinking about how fun it will be to have those simulations to validate and compare against empirical data. And once you go from there, you can go all sorts of cool places. So it's really cool, Shelly.
Thanks.
All right, I'll let Brittany if she has a question.
Well, I think Jeanette had a question. She had her hand up before I.
I'm sorry. Oh, I didn't see that. Sorry. Oh, I didn't. Jeanette, go ahead. Sorry. I had to scroll up on the chat list.
Okay, we can all step all over each other being polite. So Shelly, super ambitious model. And I wanted to ask you, like, going forward, given your question, your guiding question about distinct evolutionary trajectories, what you're thinking will come next. So are you planning to think about sort of whether or not distinct origins might interplay with, say, distinct adaptations or isolating barriers that would then set these independent origins on different trajectories? Or I guess I'm wondering about that big picture piece and how you're thinking about moving forward with that.
Yeah, that's a good question. So this is only one part of my dissertation, and I have two empirical chapters, one on Laria tridentata with Rob Lepore, Tim O'Connor, and Jacob Landis, and then another on Galix uceolata, which was originally my entire focus. And really with Galix, you have nothing that's differentiated between the cytotypes. You have the slight niche differentiation, but it's really similar. And so I think in that case, you could have these different trajectories of even ecological. So maybe microbial community composition could be, allowing persistence of different tetraploid populations. There has to be something different between these diploids and these tetraploids. And I think we're seeing a pattern that's swamped by, diversity in these different lineages, perhaps. And so I do think you could see different selection for different adaptive advantages. I don't know, the world is my oyster, I feel like, with all the things you could find. But I'm definitely interested in looking at selection and seeing if you see different strain selection or any adaptive alleles.
Cool.
All right, Brittany, and I think Shelly's cat's making an appearance. Somebody's cat.
Yeah, I wouldn't know if that was Mabel or Gus.
It's Mabel.
So at the risk of making things more complicated than they already are, do you think it would be possible to sort of, I guess, generalize the way you have these models sort of, and what Fisher did in the 40s, going from, say, tetraploids to hexaploids to octoploids? Like, is there an easy way to sort of expand those mathematics? Or are you going to have to do it sort of bespoke for each system? And, do you think it will be possible to parameterize these such that there are sort of like, common enough estimates across multiple taxa for recombination rate or, rate of tetravalent formation? Or are we going to have to do this species by species?
So I think you can do this species by species. To answer that aspect is I think that you might want to, because there is such diversity. When it comes to the ploidy level, I think that this is you might be able to use the same pathway that I use in the same bins, but those probabilities will differ. Okay. So it's something I'll have to think about more, and your matrices will just start expanding. So it depends on what you want. So if you think that it could go more than tetraallelic, I wouldn't recommend doing it in this method, because it will get increasingly more complex. The way that we're writing it should be able to be expanded over time, but it just could be computationally just not a good idea.
Totally fair.
So hopefully that answered your question.
Yeah, nice job.
All right. Any other questions for Shelly? I know there's some questions in the chat box for Wendy's talk as well. That's happening there. So yeah, I really look forward, Shelly, to seeing this. I'm just sort of very curious too. I guess my question earlier about the inheritance patterns too, it'd be fun to see how, different, if, you have sort of things diploidizing quicker, going and shifting to at least to disomic inheritance earlier, and then you get later generation origins and how those end up combining, and do those look distinctive or not genetically later on? Do they recombine well, et cetera? I'm just sort of curious how that'll play out. I look forward to seeing your talk at botany. So, all right. Are there any other questions for Wendy that didn't get addressed in the chat box already, or for Shelly? All right. Well, if not, I think we'll wrap it up here. I want to thank both Shelly and Wendy for two excellent talks. And Shelly's reminder about Botany 2021 does remind me to say, just go ahead, and I got to remember to register myself. I look forward to seeing another round of these talks there. I know there's over 1,000 abstracts submitted again this year. So it should be another really great online meeting. So go ahead and check that out if you haven't. Otherwise, we have our last Polyploidy webinar of the semester happening in 2 weeks on May 24th. Speaking at that will be Dr. Brittany Sutherland, who will talk about the population genomics of invasive Salvinia molesta in the southeastern United States, and Misha Olson, who will tell us about meiosis and neotetraploid maize. And so we should have a strong finish to the semester. We will take the summer off from the webinar series. We've ran this almost for over a year now, more or less continuously every 2 weeks, and we'll take a break and come back in the fall. So Brittany and Misha will close us out for the semester while we have a little bit of a break. But otherwise, I want to thank everyone for participating, and look forward to the last set of talks in 2 weeks. All right, see everyone then. Thank you. Bye, everybody.